Okay, hi, my name's Professor Emma Thomson. I'm consultant in infectious diseases, doctor in Greater Glasgow and Clyde, and currently based at the MRC Centre for Virus Research in Glasgow. And a part of my post is actually with the London School of Hygiene and Tropical Medicine as well, and the Uganda Virus Research Institute. So I work both in the UK and in Uganda predominantly. And my laboratory focuses on emerging viral infections and the genetics of viruses that have emerged over time. And so at the moment, we've had a very strong interest in the SARS-CoV-2 outbreak. So my laboratory stayed open throughout the outbreak, and we have been very involved in sequencing the virus. So like all living things, viruses contain within them genetic material, which is either DNA or RNA. And we're interested in sequencing the RNA, as it happens to be for the SARS-CoV-2 virus, in order to understand, for example, how the virus arrived into Scotland, how many times it arrived into Scotland. We've been able to estimate that as part of the COG UK consortium. So one of our major projects has been with the COG UK consortium, which is an initiative funded by the government, a £20 million initiative to sequence lots of viruses over time. And across the UK, we and other laboratories have sequenced now more than 43,000 genomes throughout the outbreak. So that's quite a lot of data. And we can sequence the virus really rapidly. So in the past, if you think about HIV, for example, it probably emerged in the 1920s in what was then Belgian Congo and is now the Democratic Republic of Congo. And we didn't actually identify that virus until the 1980s, until it had spread all around the world. And things are very different now. So in China, this new virus, the SARS-CoV-2 virus, emerged in November, December of 2019 and was sequenced was recognized firstly as causing a new clinical illness. And as soon as that was recognized, it was sequenced. And we can sequence viruses within 24 or 48 hours now. And this has been part of the sort of like development of technology, which has been extraordinary, so that such that we can actually sequence little bits of the DNA of a virus. We can sequence those stretches of DNA like 100 million times within 24 hours just with one machine. And that's like printing out the ACTG sequence that you see from any part of life. The genome is made up of A, C's, T's and G's. If you printed what you can sequence nowadays with the new technologies we have, it would fill up the whole room, even if you printed it on like the tiniest 6-point font size. You'd fill up the room with data if you printed it out on paper. And of course, now we have to use like really powerful computers to synthesize and put together, to jigsaw together the virus genome. But we can do it incredibly rapidly because computing has advanced massively over the last few years. So we've been doing that anyway, using that kind of genetic data to understand something about the virus and how it got around the world. We know that it jumped out of bats in China and into the human population, but we think, so some of the research that's happened at the CVR has shown that the virus probably was already in bats for the last 40 or 50 years, but jumped out very recently in 2019. And then was pre-adapted, so it was already ready to infect humans, which is quite interesting and perhaps a bit unexpected. And we know that there are probably hundreds of viruses in bats, particularly in China, that look like they might be ready to make the same jump. So it's really important that we think about why these things happen and to understand how they happen. So it's sequencing projects that look at bat samples, for example, to see what viruses there are around in the environment that might be able to jump into humans are really important. Other things that are incredibly important are that we think about the ecosystem that we live in. And as humans, we're quite an invasive species. We are chopping down forests all the time. We're eating into ecosystems which have been there for many years without causing any difficulties for us and for our health. And we know that the emergence of HIV, for example, is very likely to have been to do with changes in human activity. And similarly, that's what's happened probably with Ebola virus as well. And these coronaviruses like SARS-CoV-2 that have jumped out of bats. So it's really important when we think about sustaining our ecology without intruding on it too much. We need to think about things like population control, or we're going to see a lot more in terms of viruses coming into across our shores and into Scotland and the rest of the UK. We know from the sequence data that we have that the SARS-CoV-2 virus was introduced into Scotland at least 276 times. And that happened largely through air travel. And most of these introductions occurred through people flying back from holidays in continental Europe. And so we also, I think we have to start thinking carefully about travel and how often we travel, and not only the impact of global travel in terms of global warming, which is an increasing problem and contributes to the spread of viruses, but also the fact that we bring back viruses with us sometimes from other countries, and that we need to think carefully about how to prevent that from happening. And SARS-CoV-2 is the most recent and one of the most dramatic examples of that. Some of the other work that we're doing in terms of sequencing just now and working with the virus involves growing the virus in the laboratory and manipulating the virus. So we can do all sorts of fancy things now with the virus genome, where we can add genes that allow us to see the virus with a little gene that lights up a color, for example, in the laboratory when we're growing the virus in cells. And so at the CVR, many people have been working on looking at whether or not the virus can be cultured, whether it can infect different types of cell, for example, from different organs in the human body, and also looking at the impact of the immune response on the virus. So one of the projects that we're involved in at the moment is investigating whether or not changes in the surface of the virus, which is encoded by a gene called the spike gene. And if you look at photographs of the coronaviruses, they're named coronaviruses because they are surrounded by a sort of crown appearance, and each of the little spikes on that crown is formed by this protein called spike protein. And it's that protein that binds to the lung cells via a receptor which is called the ACE2 receptor. And so we're very interested in how over time the SARS-CoV-2 virus is changing the shape just subtly of these little spikes that come out of the virus particle. And we found a couple of changes that may be associated with slightly changed binding to that ACE receptor, and may be associated with an increase in transmission risk. Although reassuringly, we find that although we think that one or two of these mutations might be associated with a tighter binding between the virus and the cell, that isn't associated with any change in severity of infection. So at the moment, the virus looks quite stable in terms of the disease that it causes in humans. So one of the other things that we've been very involved with, that I've been involved clinically, is acting as the local principal investigator for running a clinical trial of one of the SARS-CoV-2 or the COVID-19 vaccine candidates, which comes from Oxford University, and it's called the ChAdOx1 vaccine. And so we've been looking at that vaccine, giving it to healthcare workers within NHS Greater Glasgow and Clyde to see and investigate whether or not this virus infection is prevented by the vaccine, and also to understand, even if the infection isn't completely prevented, if the vaccine can reduce the severity of infection that people get when they're infected. And we know that from studies carried out by Oxford University and colleagues of theirs, that in rhesus macaque monkeys, the vaccine can really reduce the amount of virus that's present in the lungs. But actually doesn't completely prevent you getting the virus and having it in your nose. So the monkeys, some of them still had it in their nose. And so we're very interested to see, obviously, it's really critical to see whether or not this vaccine, which is made up of the spike gene that I mentioned before. So it's a virus called an adenovirus, and it's been genetically modified or engineered. We can do all sorts of fancy things with viruses now. We can manipulate them to have the gene of the spike, which is the surface of the SARS-CoV-2 virus, and put it inside an adenovirus, which is, adenoviruses cause the common cold. And so we put this, this gene has been put in to the adenovirus, and then we inject that virus into the arm of the volunteers. And the virus itself, it produces protein, and it's that protein that the immune response recognizes. It doesn't replicate the virus, which means it doesn't divide, and so it doesn't cause an infection in itself, but your immune system, inside the cells, you will produce protein that the immune system then recognizes. And so we're very keen to understand whether or not this virus works, and several hundred healthcare workers in NHS Greater Glasgow and Clyde, I think we're now over 600, have had that vaccine. And also we've been, more recently, we have been carrying out, as part of the same study, a kind of sub-study, a sub-part of the study in people who are in an older age groups. So we know that COVID-19 infection is really, really serious once you get, it can be really serious once you get above the age of around 65. And so it's really important to test to see how well the vaccine works in that group of people. The younger you are, the less likely you are to have serious consequences of infection. And so it's really critically important that we know that we can offer it to people in the older age group as well. And so now we're recruiting people from that age group to see whether or not the vaccine works in them. And that's being carried out right across the UK. And the same study is being carried out now also in Brazil and in South Africa. So we should have an answer within the next few months on whether or not this vaccine works. And if this one doesn't work, there are more than 90 others behind it in clinical trials as well. So I think there's probably quite a good chance that we'll eventually get there with a vaccine, but of course it takes time to develop these things and to run clinical trials to make sure that they work well.